So hey guys, what's up? So welcome to World News 28 News channel. So let's talk about how virus attack step by step. It is still a novel coronavirus that causes COVID-19, but the emerging picture has given researchers clue about how to target it. A look at its structures, how it infects, and the behaviours scientists hope to block. In the search for a treatment for COVID-19 disease, researchers have been targeting specific behaviors of the novel coronavirus SARS-CoV-2 that causes the disease, while the virus itself is being studied. The hunt for a treatment is based on what is known so far about the way it infects humans. So, how does it infect someone? It begins with the spike that gives coronaviruses their name. A coronavirus is surrounded by a fatty outer layer, and on the surface of this layer is the corona. Corona of the spikes made of protein. On the surface of human cells is an enzyme called ACE2, which acts as a receptor that enables SARS-CoV-2 to launch its attack. The virus's spike protein binds to the receptor, then fuses with the cell surface and releases its genetic material RNA in the case of the SARS-CoV-2 into the cell. The coronavirus that causes SARS, called SARS-CoV, uses the same ACE2 receptor to invade a cell. Once inside, the virus replicates itself by using the cell's molecular mechanism. All these stages involve various interactions between the virus proteins and the human proteins. Any treatment being developed or research will look to inhibit these activities at one stage or the other. Which treatment specifically tries to inhibit which activity? The Solidarity Trials, a World Health Organization initiative that includes India, are investigating 4 lines of the treatment using existing drugs. Separately, various research institutions are studying the virus's functioning in the hope that the knowledge will lead to the repurposing of the existing drugs or development of new ones. The Solidarity experiments that try to find out if virus activity can be inhibited at reception stage. This is the target of the trial, which is a combination of the antimalarial drugs chloroquine and the hydroxychloroquine. Part of the hope comes from a 2005 study in the Virology journal that studied chloroquine's role against the SARS virus. It found chloroquine prevented that virus's ability to attach itself to the ACE2 receptors. However, because chloroquine causes severe side effects, the current trials are being done with a combination with its less toxic derivative hydroxychloroquine. The effect of these two drugs, SARS-CoV-2, is still being studied around the world. At cell antistrate, the chloroquine hydroxychloroquine combination comes into play again. Many viruses enter a cell by acidifying compartments within the membrane at the cell surface and then breaching at the membrane itself. When chloroquine and hydroxychloroquine enter the compartment, it loses part of its acidity. The aim of the trials is to hinder the viruses at this stage. At replication stage, a number of trials are looking at obstructing replication at a key step during which the virus is using an enzyme to break down proteins, leading to a chain of new viruses. The drug lopinavir, for example, has been known to inhibit the enzyme used by the HIV to split proteins, but because lopinavir itself tends to break down in the human body, it is used in combination with the ritonavir, which allows it to last longer. One set of Solidarity trials is looking at this combination of the anti-HIV drugs, and another is investigation lopinavir, ritonavir combined with the interferon beta, a molecule that regulates inflammation in the body. The Solidarity trials with the drug remdesivir, originally created to fight the Ebola virus, will seek to inhibit the novel coronavirus by targeting the action of a key enzyme that facilitates its replication. Previous studies had shown its effectiveness in animals infected with SARS and MERS coronavirus. This year's study, published in the Cell Research, reported that a combination of chloroquine and remdesivir can hinder replication of the SARS-CoV-2 in cultural cells. What are the other studies looking at? Some studies are looking at the structure of the virus, while others are investigating its behavior as a potential target for future drugs. For example, structure at the Max Planck Institute in Germany. Researchers identify the spike protein as not only the sharpest weapon of the virus, but also its Achilles heel. Antibodies can recognize the spike protein, bind to it, and mark it as a target for the immune cells. However, the virus also has a sugar coat that hides parts of its spike proteins from the immune cells. Therefore, the researchers are analyzing the sugar shield and trying to calculate how the spike proteins move on the surface of the virus and how they change their shape. Using supercomputers, researchers hope to identify binding sites for the antibodies and plan to compare these with the binding properties of the existing drugs, and thus identify ingredients that can block the spike protein. Of course, repurposing drugs that are already on the market is much faster than finding a new active ingredient and testing them in a lengthy clinical trial. General Human Director at the Max Planck Institute of Biophysics said in a statement. Behavior in a study last week in the Journal of the Clinical Medicine, researchers from the universities of the Bologna and the Catanzaro mapped the interactions between the virus proteins and human proteins when the virus attacks. The body responds by activating certain proteins and deactivating others to hinder it. At the same time, the body has other mechanisms that the virus exploits. These were what the researchers mapped, identifying specific proteins. This valuable information about the effects of the new coronavirus on the proteins of the human cell may prove to be fundamental in redirecting the development of the drug therapies, since compound antiviral treatments seem to be unsuccessful. That author Federico Majordi of the University of Bologna said in a statement. Beyond the solitary trials, there are studies on specific drugs. Reports are emerging from time to time in Nature last week, an international collaboration led by researchers at the Shanghai Tech University reporting 6 possible drug candidates which they identified after testing more than 10,000 compounds. The project targeting SARS-CoV-2 main enzyme for splitting proteins, MERPRO, which plays a key role in the mutating viral application. Researchers adding drugs directly to the enzyme to cell cultures growing the virus, assessing how much of each compound is required to stop the enzyme. 6 drugs appear to be effective, they reported. So if you like the channel, so kindly subscribe to it.